So for the last part of this series on cell division is that we're going to look at what happens when cell division, mitosis, and meiosis, if something goes wrong, if you have an alteration of the chromosome number and the structure. So accidents during meiosis can alter chromosome number. An abnormal chromosome count is usually from the result of what we call nondisjunction, in which there's a failure of the homologous pair to separate during meiosis 1, or for a failure of one of the sister chromatids to separate during meiosis 2. One is more devastating of a change than the other, and that, of course, is seen more so in the non-disjunction found in meiosis 1. So in meiosis 1, if there is a failure to separate of the homologous chromosome, you then will get a, basically one of the cells, one of the lines of division loses a lot of DNA material compared to the other, which gains one additional chromosome. Compare that to non-disjunction in meiosis 2, where in non-disjunction in meiosis 1, every single one of the gametes is affected. But in meiosis 2, if the non-disjunction happens, it only affects 2 of the gametes, of the 4 gametes that are possible to form. So let's talk about a karyotype. So a karyotype is what your book is calling a photographic inventory of an individual's chromosome. And basically, it's just creating a visual representation of your chromosomes. So to prepare a karyotype, you need some white blood cells. You can isolate them, create them, make them grow, and then you can arrest them at metaphase, and then you can see them underneath the microscope. And then you can take those chromosomes, which are in the what I call the traditional X shape, the condensed chromosome shape, and order them in their homologous pairs. And then you can determine if there's any chromosomal abnormalities in that picture. And if we were in a lab, you could actually do this in lab, and you could actually see your own chromosomes, and you can then arrange them and look at them underneath the microscope. Now, of course, many people don't want to take their own blood and do this process, which is why you can use other cells. For example, you can do this with your cheek cells. And so someone in a lab will do this process, and then they will create your karyotype. And so this is a representation of a karyotype, in that you will pair your homologous chromosomes. You see how they're aligned here. So you have your 2 sister chromatids and then the pair of homologous chromosomes. And humans, we have 23 pairs of chromosomes, so you can count them here, the number of the chromosome number and its pairs. There are 22 autosome chromosomes, which means they don't have anything to do with an individual sex. And then there are the sex chromosomes, X and Y. So this individual here has 1 X chromosome, 1 Y chromosome, so this would be a male individual, at least genetically. So let's make a connection to something that's real for a lot of people. An extra chromosome, or an extra copy of a chromosome on the 21st chromosome, is one of the most common forms of Down syndrome. It's trisomy 21, in that there are 3 copies of the 21st chromosome. So here is a karyotype of someone with trisomy 21. You can see they have 3 copies of the 21st chromosome. And this is, of course, the most common form of Down syndrome. There are other forms of trisomy, but this is the more common, and it's the one that doesn't really cause any huge or major differences compared to a normal karyotyped individual. Now, there is a connection to infants born with Down syndrome and the age of the mother at the time when she becomes pregnant. Uh, Women who are likely to get pregnant at a younger age, in their 20s, 30s, seem to have a lower risk of having a child with Down syndrome compared to someone in their 40s or older. As they get older, the mother gets older, the chances of the child developing Down syndrome becomes very high. So you see here that quite close to 50, there is an 80% chance or more of your infant being born with Down syndrome compared to, say, a mother of around 25, which has around a 2 or 3% chance. So trisomy is just one example. There are other examples where you can actually have changes or alterations in the sex chromosomes. And then abnormal numbers of the sex chromosomes don't usually affect survival, but they, compared to, say, some of the changes you would see in some of the autosomes, for like chromosomes number, say 1 through 5. If something was to happen, let me go back to a picture of a karyotype. If something was to happen to one of the chromosomes, say 1 through 5, any changes to these, because these chromosomes, the larger they are, the more DNA, the more genes that they have on those chromosomes. So any major changes to the larger chromosomes will lead to more severe changes in the individual. So from my knowledge and recollection, any changes to any of these large chromosomes will usually end up with low survivability. The child will be born with a lot of defects. So this is one reason why I wanted to give a discussion board on chromosomal alterations and abnormalities, because as much as we don't talk about chromosomal abnormalities, it's actually quite common, especially in parts of the world where malnutrition is higher compared to other parts of the world. And so a lot of changes or abnormalities of these larger chromosomes usually ends up in very low survivability rates, and so infant death. However, compared to the sex chromosomes, usually changing or adding numbers to the sex chromosomes doesn't really affect your survival. It does affect, though, your sexual development. So you can have an extra X chromosome or an extra Y chromosome and still end up being fine or normal. And then you can actually lose one of the sex chromosomes and still end up being a normal functioning individual. And so, for example, if you have an extra X chromosome, so an individual with 2 X's and a Y, that's called Klinefelter syndrome. It's found in male individuals. And so some of the actual physical characteristics or the problems that arise is that they can end up being sterile. They could have undeveloped testes, which usually also goes hand in hand with sterility. And they can have secondary female characteristics in that, for example, they could have breast development or or enlarged breasts when you compare them to another male that may not have enlarged or padded pectorals. You can have an extra Y chromosome, and there is not a current name for this. In the literature, sometimes you'll see these individuals be called meta males, and that they have the extra Y chromosome, and there doesn't seem to be any, at least developmental symptoms for in the sexuality or sexual development of the individuals. However, I do caution against always saying that correlation and causation are the same or go hand in hand, but there are studies trying to link an extra Y chromosome to more aggressive individuals. The triple X female, a female with an extra X chromosome. There doesn't seem to be like a name for this. A lot of times you'll see them called the meta female. There is no actual physical changes in their sexual development, and that there just seems that these individuals are just slightly taller than the average female. And then the last example here is Turner syndrome, in that where an individual only receives one X chromosome, this is literally supposed to represent a 0. So they only inherit one X chromosome. So they do display as female, but because of the only one having one X chromosome, sometimes they can be sterile individuals, and this has to deal with if their sexual organs are immature or don't fully develop. So this is very similar to what happens in Klinefelter with males who have an extra X chromosome. Now, the occurrence rates of these abnormal chromosome numbers are actually much higher than you think. Some of these occur in 1 in 1,000 males or 1 in 1,000 females. And a lot of individuals can have abnormal sex chromosome numbers and never even know it. You can go your whole entire life without ever knowing that you have an extra chromosome if it doesn't affect your development. And many times it doesn't. Just because a female is born with Turner syndrome doesn't mean they're always going to be sterile or have immature sex organs, and that's true for all of these individuals. Just because you are a male and have Klinefelter syndrome doesn't mean you're going to be sterile or have undeveloped testes. These are just the common symptoms that are seen in these individuals. So there is a connection between having abnormal or alterations in your DNA or chromosomes. So non-disjunction can produce polyploid organisms, polyploid meaning having multiple chromosome numbers. So organisms with extra sets of chromosomes. And then you can have errors in cell division, which is usually important in the evolution of new species. Mutations. So this is a gray tree frog, and this is an example of a tetraploid. So this gray tree frog is a new species that resulted from the unequal cell division of the homologous chromosomes. So this individual has 4 sets of DNA, 4 copies of the chromosomal DNA, which created a new unique species. There are many, many, many examples of chromosomal alterations that can cause birth defects and cancer. Chromosomes can undergo many different mutations or breakage. There's examples of deletions, duplications, inversions, and translocations that produce genetic disorders and then sometimes cancer. So deletion is when parts of a chromosome have just been deleted. During synthesis, when these were being copied, for some reason an area was skipped or not copied. You can have duplication in that the opposite problem can occur, in that an extra part has been copied twice. And then you can have inversion, where parts of the chromosomes can actually be inverted. No clear reason why that can happen. But remember, synthesis of the new DNA, copying new DNA, is such a complicated process that you shouldn't be surprised by any mistake that occurs. And this is the most damaging would be reciprocal translocation, where literally two parts of two separate chromosomes will swap places and create chromosomes that where the DNA and loci of non-homologous chromosomes are now in the wrong place, the wrong spot, and so your body doesn't recognize or know where those genes are located. So an example of how this can affect actual, humans in real life is translocation of chromosome 9 and 22, and that these two parts of the chromosomes will be translocated into the wrong spots after the copying here, and this creates a cancer-causing gene in this chromosome, which will lead to one of the forms of leukemia. There are many examples of how these DNA chromosomal alterations can lead to many birth defects or cancers or characteristics that are not ideal and creates problems for individuals, where many of them can be life-threatening or lead to death.